How's everything going this morning?
Pretty good.
Had a flat on the way to school today. That was interesting.
It's not fun.
Yeah, because it got cold, all my low tire indicators were going off. So I went to the gas station to fill it up. And as I was filling up the last tire, when I pulled the inflator off of it, it broke the stem. And so I just sat there and watched all the air I just put into the tire flow out. And yeah, that wasn't so good. Anyway, I got it changed and got it to the shop and got it to school on time. So that's all that really matters. So you guys have been at this for a week and a half. How's everything going?
Good.
Adjusting, all right?
Slowly.
Yeah,
Slowly?
doing my best.
Okay, well, that's all we can ask. All right, well, it's 9 o'clock. You guys have any questions over what we were going over last time? We're kind of in the middle of photosynthesis, so you might have more questions at the end of today. All right, then let's get started, I guess. Give me a second to share a screen with you.
Okay.
Okay, can you guys see the photosynthesis slides?
Mm-hmm.
All right. Is it just the full screen, or can you see the presenter view?
You can see the
You see just
presenter
one?
view.
Oh, you're seeing presenter view. All right. Now what are you seeing?
Full screen.
Okay, cool. Okay, so we started out kind of looking at leaf structures last time. Recall that the only part of the leaf structure that you really need to know about is the mesophyll cells, the vascular bundle, which is surrounded by these bundle sheath cells, the spongy mesophyll in particular, the stoma and the guard cells that guard the stoma. These are all the parts of the leaf that are heavily involved in photosynthesis. So they're really the parts of the leaf that I want you to be able to identify if on the exam you see an image of the leaf and you're asked to label parts of it. And I always put on exams the images of the leaf that were in the PowerPoint. So I'm not going to pull up some weird wonky version of a leaf that you haven't already seen. And I do that with all the figures for the most part on exams. So last time we started on photosynthesis, and I gave you the equation. Carbon dioxide and water gets converted into glucose, and it has oxygen and water as byproducts. You need energy from the sun, energy from light, and chlorophyll and rubisco in order to facilitate this. The chlorophyll captures the energy from the sun. The rubisco is the enzyme that is involved in carbon capture.
Are we gonna have to know that formula?
No, what you need to know is that you start off with carbon dioxide and water, and you end up with glucose with a couple of byproducts. I'm not going to ask you to write the equation out, but you should know at least the names of the beginning reactants and the products that you get at the end. So I won't ever ask you to recall it, but I will ask you things about it. Does that make sense? Who was that?
Blair.
Asking a question. Blair, okay.
Yep.
So one thing that I will want you to know, though, is I want you to know the name of ribulose bisphosphate carboxylase oxygenase. So Rubisco is this kind of acronym. And last time, towards the end of class, we were talking about why it's called this. And so it's called this because Rubis stands for ribulose bisphosphate. That's the substrate that then the enzyme adds either a carbon dioxide to or an oxygen to. So the CO is for the carboxylase function and the oxygenase function. So you can think of this as a Swiss Army knife with two blades in a sense. One blade adds carboxylase, the other blade adds oxygenase, and those are the only two functions that it has. Most enzymes have a single function, but in some cases, enzymes have multiple functions, and this just happens to be the case of one that has multiple functions. It adds carbon dioxide to ribulose bisphosphate much more easily than it adds oxygen. But if all it has to work with is oxygen, it'll just add oxygen. And so you'll see why this becomes important later on. All plants use Rubisco. And so whether you're a C3 plant or a C4 plant, you use Rubisco for this initial carbon capture phase of photosynthesis. In C3 plants, you add a single carbon dioxide to the 5-carbon ribulose bisphosphate, ribulose bisphosphate, which is a 5-carbon molecule. You add a carbon dioxide to it, that converts it into a 6-carbon molecule, but that 6-carbon molecule breaks down very quickly into 2 3-carbon molecules. And so the first stable intermediate product has 3 carbons, which is why this is called C3 photosynthesis. It occurs in mesophyll cells, and it occurs in plants like wheat and soybeans. So this is all just review from last time. We also have another type of photosynthesis called C4 photosynthesis. In C4 photosynthesis, the first intermediate product has 4 carbons rather than 3, thus C4 photosynthesis. This occurs in both the mesophyll cells and the bundle sheath cells, and the spatial division actually is important, and we'll see why here in a little while. It uses chlorophyll and Rubisco, but there's another enzyme that it uses called PEP carboxylase. And so PEP carboxylase in C4 photosynthesis is responsible for initially capturing carbon dioxide. You'll notice that PEP carboxylase is just a carboxylase. It's one of the enzymes that only does one thing. And what it does is it adds a carbon dioxide molecule. That's what carboxylase means. It adds a carbon dioxide molecule to a compound called phosphoenol pyruvate. And we'll just shorten it to PEP. PEP is a 3-carbon compound. When you add a carbon dioxide to it, it becomes a 4-carbon compound, and that's why this is C4 photosynthesis. And this occurs in C4 plants, and agricultural crops that are good examples of C4 plants are corn and sugarcane. So these are the two most common photosynthetic pathways: C3 photosynthesis and C4 photosynthesis. And this is just some basic introductory information about those two forms of photosynthesis. We're about to get into the weeds of it here in a few minutes. Questions at this point? Okay. You'll recall from last time that I talked about photosynthesis occurring in 2 phases: the light-dependent reactions and the light-independent reactions. And so I want to briefly introduce you to these 2 types of reactions. So in the light-dependent reactions, rather, these are the reactions where you actually have to have light present. And the reason that these are important is because the capture of light by the chlorophyll is basically where we get all of the energy for building the molecules of glucose that are going to be built through the process of photosynthesis. So you see in the light-dependent reactions, there's a lot of capture of energy from light. It excites electrons, drives electrons away from molecules that are inside chlorophyll, and then it harvests the energy that those energized electrons got from the light to basically create ATP and NADPH. ATP and NADPH are basically, ATP is essentially a compound that stores energy. It's one of the energy storage mechanisms that cells have. And then NADPH carries around hydrogens. And as it's carrying around hydrogens, it can set up diffusion gradients. And then those diffusion gradients can be used to generate energy in cells. And so these are basically two ways in which you can kind of store energy, either storing energy in the bonds of a molecule. So phosphorus molecules are bonded together with triple bonds, and those triple bonds take a lot of energy to break. They also take a lot of energy to form. And so by stripping off a phosphorus molecule from an ATP, you release a ton of energy. Or you can also generate energy through moving substances around in cells. And so the light-dependent reactions, you don't really need to know much about them, except that this is where all the energy comes from. Now that energy is going to be used in the light-independent reactions, and this is where we're going to spend most of our time. It uses ATP and it uses NADPH in order to take this 5-carbon ribulose bisphosphate and go through a number of cycles of this cycle to convert carbon dioxide into sugar, essentially. And so we're going to be spending most of our time on this cycle to the right, and we're going to spend it especially at this initial phase when carbon dioxide gets captured by the cells. So know that the light-dependent reactions are where the energy is coming from, but that energy that is harvested in the light-dependent reactions gets stored in chemical bonds in the light-independent reactions. Is that clear? Okay. So remember that the mesophyll cells is where this is going to be happening for the most part, except in C4 plants where it's going to be occurring in both the bundle sheath cells and the mesophyll cells. So this is an animation. So when you download the slides from the Moodle site, it's a good idea to download them as PowerPoint slides and view them in PowerPoint. If you don't have PowerPoint on your computer or your iPad, you can get that free from the school. Contact a guy named Keith Hayes. He will tell you how to do that if you haven't done that in the past. The reason it's useful to do things in PowerPoint is because this slide is animated, and it's animated to drive home the point of how the process of photosynthesis operates, okay? And if you just download it as a PDF or something, that flattens all the animations, and so you just don't have as useful of a study aid if you do it that way. So here's our leaf. It's just the same figure here, but we're just looking at kind of a two-dimensional slice through it. Here we have the upper epidermis, the lower epidermis. We have all of the mesophyll cells. We have the bundle sheath cells that go around the vascular bundle. I've put rubisco here because rubisco resides inside the mesophyll cells. So that's where you find this enzyme. And then you can see that there are two guard cells up here at the top. And then I've covered up these guard cells down here at the bottom with little circles that represent the guard cells with the stoma closed. And then this open space here and this open space up here represents the air spaces that are between the mesophyll cells that you see in this image here. You see how there's all this air space down in the lower portions of the leaf. So the stoma is this opening that basically connects the air spaces inside the leaf to the air outside of the leaf. And so when the stoma are closed, you have carbon dioxide, you have oxygen, and you have water, and those are all at certain concentrations. And at the beginning of photosynthesis, the amount of carbon dioxide and the amount of oxygen that are in the air spaces are similar in terms of their concentration. Now, in the atmosphere, there is a lot of nitrogen, but the next most common thing in the atmosphere is oxygen. And carbon dioxide actually forms a really small portion of atmospheric air. So the concentration of carbon dioxide is really low. The concentration of oxygen is relatively high. And then water varies because this airspace is now closed up. It's receiving water vapor from the surrounding cells. So the concentration of water is much higher inside the leaf than it is outside of the leaf, which is why the water inside the leaf is in dark font, and the water outside the leaf is in a gray font to represent that there's more water, a higher concentration of water in the leaf than out of the leaf. Carbon dioxide and oxygen look the same because they're at similar concentrations inside the leaf and outside the leaf. So that explains kind of why things are written the way they are in this figure. Now, what happens when light starts hitting this leaf and it starts undergoing the process of photosynthesis is it begins taking carbon dioxide, capturing it, and converting it into glucose. So over time, the glucose gets absorbed by the cells and gets bound using Rubisco to ribulose bisphosphate and gets converted to glucose. As that happens, what happens to the concentration of carbon dioxide in these air spaces? Carbon dioxide gets converted to glucose. What happens to the availability of carbon dioxide?
It's down.
What's that?
It goes down.
Yeah, it'll go down. So over time, you will see that carbon dioxide goes down. And it goes down eventually to the point where carbon dioxide levels are really, really low. And here's where it gets complicated. As carbon dioxide concentration goes down, because Rubisco can also grab oxygen, you risk grabbing oxygen, and that inhibits your ability to make glucose. So as carbon dioxide concentrations go down, oxygen has the possibility of interacting with Rubisco, and the plant doesn't want that to happen. So what the plant has to do is the plant has to open up its stomata. Those guard cells open up the stoma. When it does that, carbon dioxide flows in and it restores the concentration of carbon dioxide inside the plant, because you're going from what was a high concentration of carbon dioxide outside the plant and a low concentration of carbon dioxide inside the leaf. And substances generally flow down diffusion gradients. So you have carbon dioxide moving to a place where there isn't much carbon dioxide. But that's not the only diffusion gradient that exists here once this is open. What other diffusion gradient exists?
H2O.
Yeah, water. There's a high concentration of water inside the cell, a low concentration of water outside the cell. So which way is water going to flow? Somebody other than Blair. Tired of hearing from Blair. I'm not, but give somebody else a chance to chime in.
Outside the cell.
Yeah, it's going to flow from inside the cell to the outside of the cell. So the whole time that the plant keeps its stomata open, water is going to flow out of the leaf. And so it behooves a plant to only open its stomata as often and as long as it absolutely needs to, because while opening it helps the plant get more CO2, in the process of doing that, it also loses water. So the reason for this, the reason I want you to view these in PowerPoint is because the animation actually kind of illustrates the point. During photosynthesis, rubisco captures carbon dioxide and converts it ultimately into glucose. As that happens, the concentration of carbon dioxide goes down. When the leaf then needs more carbon dioxide, it opens its stomata. When it opens its stomata, carbon dioxide flows in, but water flows out. Is that clear?
Yeah.
Okay, all right. So if you live in a cool environment with a moist climate, opening and closing your stomata doesn't really matter, because if you lose some water in the process of opening your stomata to restore carbon dioxide, it's not a big deal because there's plenty of water in the atmosphere, so the diffusion gradient isn't super huge. And there's also plenty of water in the soil so that you can always replenish that water supply by absorbing water through the roots and pumping it up into your leaves. But if you live in a hot, dry environment, anytime you open your stomata to let carbon dioxide in, you risk losing water. You're going to lose more water because the diffusion gradient is higher, and you aren't going to be able to replenish that water very well from the roots because you live in a hot, dry environment. And so species that live in hot, dry environments evolved another way of going about doing photosynthesis. And so this is fundamentally the difference between C3 photosynthesis and C4 photosynthesis. In C3 photosynthesis, all of the photosynthesis goes on in the mesophyll cells. But in C4 photosynthesis, initial carbon capture goes on in the mesophyll cells, but the actual manufacture of glucose goes on in the bundle sheath cells. And the bundle sheath cells are interesting because they have a coating around them, a thick wall around them that is impregnated with lignin. And the thing about lignin is that it is relatively impermeable to gases flowing through it. And so I'm going to go back and pull some stuff up from earlier. Let's go back. Oh, I'm going to have to go back to the previous. When we go back to plant structure, and I don't have any figures of this. I should put a figure of this in here. I need to give you, well, maybe it's on the, let's go back to the other one. Let's go back to photosynthesis. Oh, I inadvertently closed the photosynthesis file. There it is. Okay, here we go. All right, why does Zoom disappear when I do this? You guys are still with me, right?
Yes.
Hello? Okay, all right, good. Yeah, I was having this difficulty yesterday during some meetings. It wants to get rid of Zoom when I play the slides. All right, well, disappointing. Okay, can you see the slides now?
Yes.
Okay. So here's an actual microscopic image of a leaf. And so here's the vascular bundle, and this represents the bundle sheath cells. And it's a little hard to see, but one of the things that you'll notice is that these bundle sheath cells have really, really thick walls. And the reason for this thickness is that they are thicker, and they're thicker because they're surrounded by lignin. And what lignin does is it essentially forms a barrier that makes it more difficult for things to flow into and out of the cells. And the reason that that's important is because one of the things that the plant is attempting to do in doing this is basically protect those cells from gases flowing into those cells. And so out here in the mesophyll, let's go back a slide. Out here in the mesophyll cells are exposed to all of these air spaces. Well, the bundle sheath cells are also exposed to all those spaces as well, except that they have this protective coating around them. What that means is that the mesophyll cells are exposed to both carbon dioxide and oxygen, but because the bundle sheath cells are protected, they're not exposed to either carbon dioxide or oxygen. So in C4 plants, what they've done is they have essentially divided photosynthesis up into two different sections spatially. One part of photosynthesis goes on in the mesophyll cell, and the other part of photosynthesis goes on in the bundle sheath cell. And so what happens is that out here in the mesophyll cell, carbon dioxide gets captured, but it doesn't get captured by rubisco. It gets captured by PEP carboxylase. PEP carboxylase is the enzyme that adds carbon dioxide to PEP. PEP is a 3-carbon compound. When you add carbon dioxide to it, it becomes a 4-carbon compound called oxaloacetic acid. And then what happens is that new molecule gets transported into the bundle sheath cells through the plasmodesmata that we talked about last time, or 2 times ago, actually. I'm going to escape, hopefully going to escape if I can, this slideshow so that I can pull up the plant structure slides that we looked at last time. When we were talking about cells, I told you about these plasmodesma, these connections. Can you see the plant cell slide? No, you can't. Can you see the plant cell slide? No?
Yeah.
Remember that there are these channels that go from the cytoplasm of one cell to the cytoplasm of another cell through the cell wall. So these connections between cells allow cells to communicate with one another, but they also allow cells to transport substances from one cell to another. Well, this is exactly what's going to be happening in this situation here, where you have a substance being created in the mesophyll cells and then being transported into the bundle sheath cells. So PEP gets carbon dioxide as a result of the action of PEP carboxylase enzyme. This 4-carbon molecule then gets transported into the bundle sheath cell, and as soon as it gets transported into the bundle sheath cell, it strips off the carbon dioxide and gives that carbon dioxide to rubisco. So here's a crucial difference between C3 and C4 photosynthesis. there is no Rubisco in the mesophyll cells. As a result, Rubisco never comes into contact with oxygen because the mesophyll cells are in contact with oxygen, and the cell wall around the bundle sheath cells protects that cell from both carbon dioxide and oxygen. So in order to get the carbon dioxide in there so that Rubisco can operate on it, you have to capture it, add it to a molecule, and then transport that molecule inside. Once that molecule is inside, the CO2 that was added to it comes off, interacts with Rubisco, and ultimately makes glucose. Because of this, Rubisco never comes into contact with oxygen in here. It only comes into contact with carbon dioxide. So because of that, oxygen never interacts with Rubisco, and so you never get the possibility of diverting this cycle that produces glucose. It never gets diverted because Rubisco never interacts with oxygen. In C3 plants, because all of this goes on in the mesophyll cells, carbon dioxide and oxygen are competing with one another for access to Rubisco. What that means, so this is another way of thinking about it. In C3 plants, carbon dioxide is captured by Rubisco, added to this 5-carbon compound ribulose bisphosphate, and then goes through the ribulose bisphosphate is over here, gets converted to PGA, which ultimately gets converted into glucose. You don't have to worry about PGA. You just need to know that Rubisco captures carbon dioxide and gets converted into glucose. In the C4 plants, carbon dioxide gets captured by PEP carboxylase, converted into this 4-carbon oxaloacetate, which then gets transported into the bundle sheath cell. At that point, carbon dioxide is freed. Carbon dioxide is captured by Rubisco and then gets converted into glucose. So the bundle sheath cell and the mesophyll cell in C3 plants are essentially doing the same thing. The only difference between C3 and C4 plants is this initial stage of carbon capture. And the reason that we do the carbon capture this way in C4 plants is because PEP carboxylase only interacts with carbon dioxide. It's only a carboxylase. It's not a carboxylase oxygenase. As a result, it doesn't ever have to worry about depleting carbon dioxide out here. So what can happen in a C4 plant is that as C4 photosynthesis goes on, the carbon dioxide gets captured not by Rubisco, but by PEP carboxylase, which means that as the carbon dioxide gets depleted, that cell doesn't need to open up its stomata because PEP carboxylase isn't going to interact with any other molecules that are inside of here. So it can deplete carbon dioxide all the way down to nothing before it finally opens up its stomata, because oxygen doesn't interact with PEP carboxylase. Oxygen only interacts with Rubisco. As a result, once it does have to open up its stomata, it can open them up very quickly for a short period of time because the difference in concentration between carbon dioxide outside the leaf and inside the leaf is really high. And so this process of opening the stomata and carbon dioxide flowing in, that's a very quick flow of carbon dioxide in because the concentrations are very different. And so it doesn't have to keep its stomata open very long, which then reduces the amount of water that leaves the leaf. And so this spatial separation allows C4 plants to fully utilize all the carbon dioxide that is in their leaf spaces before it finally opens the stomata. The stomata don't have to stay open for very long, which then conserves water. So this is a water conservation strategy that some groups of plants have arrived at over the course of evolution. Does that make sense to you? Or is it way too complicated? Everybody unmute yourselves for a minute. I just loaded a bunch of science on you, and I need to know how you're dealing with it.
What'd you say the main difference was? Kind of just how it gets captured, right?
That initial capture of carbon. So in C3 plants, it happens in the mesophyll cells. So one way of thinking about this, and there are a variety of ways, and I'm going to show you a variety of ways. One way of thinking about this is to make a table. So C3 and C4 plants differ in a number of ways in terms of initial carbon capture. One way is where it happens. In C3 plants, where does it happen? Initial carbon capture.
Rubisco.
Well, and what kinds of cells?
Mesophyll cells.
The mesophyll cells. Where does initial carbon capture happen in C4 plants?
Bundle sheet.
Not initial carbon capture.
It's also the mesophyll.
Mesophyll.
It's also the mesophyll cells. Okay, so there's no difference here. What is different about the mesophyll cells, though, is what enzyme is being used. What enzyme is being used in the mesophyll cells of C3 plants?
Rubisco.
Rubisco. What's being used in C4 plants?
F.
PEP carboxylase. Now, because you're using different enzymes, you end up adding carbon dioxide to, in this case, ribulose bisphosphate. In this case, you are adding it to PEP. And the other thing is, once you've done this, glucose is formed in which cell?
C3.
In C3 plants.
Mesophyll.
It's formed in the mesophyll cells. Where is it formed in the C4 plants?
Bundle sheet.
That's where the bundle sheath cells come in. So at some point, you have to get that carbon dioxide from the mesophyll cell into the bundle sheath cell. And how do we do that? Well, we do that by adding carbon dioxide to PEP, then transporting that from the mesophyll cell to the bundle sheath cell. So one way of dealing with the complexity is to just put it in a table and say, in what way are these two processes similar and in what way are they different? And does that help at least some of you?
Yep.
Yeah.
Here's another way that might be useful to you. You guys know what an analogy is?
Yes.
What's an analogy?
It's like a metaphor or similar, referencing something through a similar situation.
Yeah, so it's basically this thing is like this other thing. And if the first thing is easier to remember, it helps you remember the second thing. So C4 photosynthesis. Is like Rapunzel. You guys remember Rapunzel from your childhood? Who was Rapunzel?
She was
Long hair.
locked up in a tower with long hair.
She was a beautiful princess. She was sequestered in a tall tower. And what was planted at the bottom of the tower?
I don't remember. I didn't know there was anything at the tower.
A bunch of spiky plants with thorns all over them were planted around the base of the tower. And the spiky plants were there. They blinded anybody who tried to go to the tower. And so as a result, Rapunzel was protected from any suitors, any princes who wanted to have romantic relations with Rapunzel, basically. And so what did Rapunzel do? She grew long locks of hair that went out the window and down so that the handsome prince could climb her hair and get access to Rapunzel. So in what way is C4 photosynthesis like Rapunzel? Rapunzel is basically Rubisco in a C4 plant. And Rubisco is in the bundle sheath cells, which are kind of like the tower. And the bundle sheath cells are protected down here by the lignin in the cell walls of the bundle sheath cells. So Rubisco is found in the tower of the bundle sheath cells, protected because of the structure of the bundle sheath cells. What is it being protected from? What do you want to protect Rubisco from that competes for access to Rubisco?
Oxygen.
The oxygen.
Yeah, you want to protect it from oxygen, but you don't want to protect it from all things, because ultimately Rapunzel wants to be with the handsome prince. In this case, who is the handsome prince?
Carbon dioxide.
The handsome prince is carbon dioxide. And what does the C4 plant equip Rapunzel with to allow carbon dioxide to finally gain access to Rapunzel, the Rubisco?
Would it be PEP?
PEP carboxylase. PEP carboxylase is the access that helps you get carbon dioxide out of the mesophyll cell. So this is now the mesophyll cell, is the wide world, not the tower. PEP carboxylase captures the carbon dioxide and allows it to then make it into the tower to get access to Rubisco. An analogy that kind of tells the story of what's happening in C4 photosynthesis. Mesophyll cells is where PEP carboxylase captures CO2, transports it to the tower where Rubisco lives. That tower is protected from other gases, and that tower is the bundle sheath cell. And then once the carbon dioxide can interact with Rubisco here, you have a romantic marriage made in heaven, and you can use that carbon dioxide to make as much glucose as you can. So that's just another way of thinking about this in a storytelling sort of way. I don't know if that was of any use to you or not. Sometimes people are like, that's a pretty strange analogy, but I don't know. I try and come up with as many ways of remembering stuff as I can. Are you following the process? Okay, all right, well, that's what I need you to do. Okay, let's go back to sharing the screen. Okay, back on the C3 and C4 plant difference. Can you see this screen?
Yeah.
Okay, cool. So what happens when oxygen interferes with the process of carbon fixation is normally carbon dioxide gets bonded to ribulose bisphosphate using Rubisco, creates some intermediate molecules, these 3 carbon molecules, and ultimately generates a carbohydrate. It goes through 6 cycles of this process, and after it's gone through 6 cycles, you have enough carbohydrate to make a molecule of glucose, which has 6 carbons. But what happens when oxygen binds to Rubisco, it forms 2 molecules that are different. And as a result, it has to go through this cycle more often, using more ATP and NADPH. As a result of that, it still makes glucose ultimately, because you still have some of that intermediate, but rather than having 2 3-carbon compounds, you only have 1 3-carbon compound that gets converted into glucose. So you have to go through this cycle more times, using more energy that has been created by the light reactions. So you eventually get glucose made. It just takes a lot more energy in order to do it. And so what plants need to be doing is they need to be maintaining carbon dioxide concentrations in their intercellular spaces in their leaf at levels high enough that oxygen doesn't start competing for access to rubisco with carbon dioxide. Carbon dioxide needs access to rubisco to avoid what we refer to as photorespiration. Photorespiration is another pathway for making glucose. It's just really energy inefficient. The normal Calvin cycle, using CO2 to bind to rubisco to create glucose, is a much more energy efficient way of doing it. Takes fewer turns of the cycle, and each turn uses less energy. And so for this reason, plants want to either be in an environment where they can open their stomata whenever they want to get more CO2 in, or they had to evolve a strategy to allow them to open their stomata less often if they occur in hot, dry environments, because you don't want oxygen interacting with rubisco. Is that clear? Okay. So once again, here's another way of thinking about it. C3 photosynthesis, it's favored in cooler climates. Photorespiration occurs whenever temperatures get high or carbon dioxide concentrations get low. C4 photosynthesis, favored in warmer climates. It avoids photorespiration and the extra energetic costs, but it does have some extra energetic costs at low temps and high CO2 concentrations. So when we go back and think about this process, there's energy being used up here in addition to the energy that is normally just used to go through normal carbon fixation and glucose building. So there is some extra energy used up here, but it's less extra energy than is used up in photorespiration. So it ends up being more efficient under hot, dry conditions than C3 photosynthesis, but C3 is the most energy efficient under the right conditions, cool, moist conditions. So this is just a figure showing the daytime growing temperature. As temperature goes up, C4 grasses are favored, but as temperature goes up, if you increase carbon dioxide concentration, C3 grasses can be favored at low temperatures and high concentrations of CO2. C4 grasses are favored at high temperatures and low concentrations of carbon dioxide. Another way of thinking about this is to look at what proportion of grasses are actually C4 and C3 grasses. So this is a figure from Argentina, where you go from the coast up into the Andes. Down on the coast, it's basically a desert. And in the desert, C4 grasses dominate. As you go up in elevation, it gets cooler and moister. And as you go up in elevation from site 1 to site 2 to site 3 to site 4 to site 5, you basically go from C4 grasses dominating to a place at these middle elevations where there's a mixture of C3 and C4 grasses, to a higher elevation where it's cooler and moister, where C3 grasses are dominating and there are hardly any C4 grasses. This is in Argentina. You can go to Hawaii and look at a similar thing on the slopes of Mauna Loa. And once again, at low elevation, you see C4 grasses dominate. At these middle elevations, you see a mixture of the two, and at high elevations, you see C3 grasses dominating. So this is a common pattern that you see throughout the world in terms of the relative abundance of C3 and C4 grasses. You see C4 grasses in hotter, drier environments, C3 grasses in cooler, moister environments. I realize you're going to think that I'm now kicking a dead horse, but if you look at photosynthetic rate and leaf temperature, photosynthetic rate is highest for C3 plants at low leaf temperatures, and it's highest for C4 plants at high temperatures. I'm going to kick the horse again, and what you will see is that if you look at the relative abundance of C3 and C4 grasses, this is the percentage of the plant community that is C4. Down in the south of southern United States and northern Mexico, 80% of the plants are, 80% of the grasses are C4 grasses. As you move north and get into cooler and cooler climates, the proportion of grasses that are C4 grasses goes down until you get up into Canada, where you cannot find any C4 grasses at all. And so climate allows for C3 grasses to dominate in cool, moist environments and C4 grasses to dominate in hot, dry environments. And it's all due to this spatial separation of initial carbon capture and where glucose actually gets created. A map, I'm going to kick the horse some more. A map of C3 and C4 grasses. C3 grasses occur northern and in the montane west. C4 grasses are in the south and in the Great Plains, where it's generally kind of on the hot, dry side. And to beat the horse a little bit more, this is a map of wheat plantings in the United States. So these are data from the Department of Agriculture, basically showing the number of acres in each county throughout the United States. So each of these little boxes is a county, and it's showing where wheat is being grown. And there's a lot of wheat grown up in Montana and North Dakota. There's also a lot of wheat grown in western Kansas, which you would think of western Kansas as being hot and dry. But there's something special that happens in western Kansas, western Oklahoma, and northwest Texas that allows you to grow wheat there. When you look at corn, you see that corn is grown in the Midwest, upper Midwest, once again, hotter and drier than you would be up at the Canadian border. So what's different about western Kansas? I'm going to go to my computer and I'm going to pull up Google Earth. Do you guys use Google Earth for anything? You guys ever used Google Earth?
Not really.
Not really? So Google Earth is a cool program. I end up using it a lot for work, for field work and whatnot. And the cool thing about it is that you can zoom in anywhere on Earth, literally. So here we are in the Kansas City area. Here's Kansas. And remember, I told you that there's a lot of wheat that is being grown up in Montana and North Dakota, but then there's this kind of hot, dry section of Kansas where you wouldn't necessarily expect wheat to thrive, but actually it does. There's a lot of wheat production in this part of Kansas, where you would normally expect kind of to be dominated by corn. So let's go and look and zoom in on this section of Kansas. As you start to zoom in, what do you start to notice?
There's a lot of green.
There's a lot of green, but what's the shape of this green?
They're in squares.
There's also a few
There-
circles though too.
Yeah, they're all circles. What is up with all these little round patches of green in western Kansas? This is basically the wheat producing section of western Kansas. Some of the circles are really small, some of them are bigger. Zoom in on them. This is a wheat field in western Kansas. What is this representing? Do you guys know? You'll see these occasionally as you're driving down the highway. You see them, especially if you drive out towards Lawrence. There's a whole, as you get to the Kaw River Valley, there are a bunch of these that you can see on both sides of the road. This is what we call center pivot irrigation. So there's a pipe coming up out of the ground right here in the middle of this thing. And there's a long pipe that goes out here and it has sprinklers all around it. It's got wheels and it basically goes around and around and around and around in a circle all day long. And it's basically sprinkling the circle with water. And so the reason that you see these circles of green, you see each of them has a line. That line represents where the arm is that has the sprinklers on it. It's going around and around in a circle, watering the crops. Well, in this section of Kansas where you see all of these green circles, there's a large aquifer called the Ogallala Aquifer, that underlies this region of western Kansas, western Oklahoma, and extreme northwestern Texas. It's probably the largest aquifer in the United States. And all of these center pivot irrigation systems in this part of Kansas are basically drilling down and tapping that aquifer, pumping that water up, and increasing the amount of water that is available for growing crops here. And so the only reason that you have this pattern, the only reason that you have this pattern for wheat in the western US is the fact that this little patch of green right here is all being artificially supplemented with water from an underground aquifer, underground water storage that has been there for tens of thousands of years. Otherwise, you wouldn't probably see much wheat being produced around here at all. You would see it mainly up here in the cooler parts of the United States. You also see that there's a little smattering of wheat production right along the Mississippi River. Once again, a place where you can irrigate effectively. You see corn in areas where irrigation is not as readily available, and you see that it's not necessarily connected to water sources like the Mississippi River. Mississippi River is right here, but you see corn production all throughout Iowa, all throughout Nebraska, all throughout Illinois, regardless of the presence of rivers. So when we ask ourselves why we grow so much corn and soybeans, or corn and wheat for that matter, and where we grow corn and soybeans, a lot of it has to do with the fact that wheat and soybeans are C3 plants and corn is C4 plants. And we grow those because there are sections of the United States where growing those is pretty easy because the climate is suitable for, in some cases, a C3 plant and in other cases, a C4 plant. Now, our whole purpose of going down this path was the second section of chapter 1, the section called Corn Walking. Michael Pollan talks about the fact that C4 plants do photosynthesis in a different way than C3 plants do. And as a result, the carbon isotopes that C4 plants collect and incorporate into glucose is different than what C3 plants do. And the reason is that C4 plants use up all of the carbon dioxide in their leaves before they open their stomata. And lighter isotopes, C12, are easier to incorporate into glucose because they weigh a little bit less than the heavier isotopes of carbon. And so C4 plants use C12, C13, and C14. So they use not only the light isotope of carbon, but they also use heavier isotopes of carbon, whereas C3 plants don't use the heavier isotopes of carbon because when they get low on carbon, they just open their stomata and let more carbon dioxide in. And so in order to understand that section of Michael Pollan's book, it's useful to know something about the actual differences between C3 and C4 photosynthesis. So let's have a little quiz here. So this is just, there is no quiz here. So because the corn is a C4 grass, it thrives in warm, dry ecosystems without needing irrigation. So the corn belt is basically what that describes. Wheat is not as well suited to the corn belt and is grown farther north and west, but remember that this growth in the west is mainly due to supplemental water happening due to this large aquifer. And people are actually concerned about the levels of that aquifer because over decades of use for things like growing wheat in a place where wheat probably shouldn't be grown, the aquifer has been drawn down quite a lot, and they're worried that there's going to come a point where it's not going to be high enough that the current wells can access it to. So there's going to be a need for drilling of deeper wells in order to tap the lower portions of the aquifer if you're going to keep relying on it for wheat production. Questions about C3 and C4 photosynthesis? All right. Did you guys get the email about the Socrative pretest? Okay. So just be sure and take that in the next couple of days. It'll come down at the end of the day on Sunday. So just carve out time. I think it's 50 questions if I recall. So it shouldn't take you that long to do, but get it done. Once again, I would have given it to you earlier, but Socrative only allows me to run one exam at a time. So I had an exam running in my other class. But do that before you gain too much more knowledge because there are photosynthesis questions on the pretest. So, is anybody else having problems with WordPress and making posts?
I just had a question about our food journals.
Oh yeah. Mm-hmm.
Are those due today, or how we turn those in?
No, you turn them in every 2 weeks. So the 2-week period for blogging is the same as the 2-week period for journaling.
Okay, and so for the journals, do we bring them to your office or email them, or?
Yeah, if you're keeping them in a notebook, in a handwritten notebook, just drop them off at my office on Friday morning of the next week. And if you're doing it electronically, just send it to me electronically on Friday morning of next week.
Okay, and how detailed do you want it? Do you just like want what we ate, just like write it down?
Drink. Yeah, so, what'd you have for breakfast? I had today I had nothing because I got messed up with a flat tire. But let's say that today I stopped at Quick Trip and got a ham and Swiss and egg croissant and mochaccino or something. I don't know what do you get at Quick Trip. Or if you go to the cafe and get a breakfast burrito or whatever. Well, what was in the breakfast burrito? Was it just eggs? Was it eggs and peppers and sausage? You got hungry mid-morning and you had a Snickers bar. Put the Snickers bar in there. Yep. So yeah, detailed. What did you eat? What did you drink?
Okay.
And just keep up with that on a daily basis. You won't do it next weekend because I'll have your food journal in my possession. But then on Monday, I'll set them back outside my door and you can come by and pick them up on Monday. What else? All right. Well, enjoy your long weekend, and I'll see you on Wednesday.